Okay, good morning, everybody. Hope you had a good weekend. Forgive me if I'm moving a little slow this morning. 3 of my 4 kids are into lacrosse, and I was at 5 lacrosse games all over the Bay Area in the hot weather this weekend, and keeping sport most of the games. So it was fun, but tiring, as will always happen with kids. Ask your parents. Office hours today, Monday, noon. Today's Monday, noon to 1. Thursday, 2:10 to 3 in 2084 BLSB. If you do not have a good understanding of negative feedback, why it maintains consistency, you should try to attend one of those office hours. It's a very important concept. I just realized there's about 2 weeks left, right, Mike? And I have that, when's that Q&A session? Wednesday, May 6th. Is it May 6th? Yeah. 8th? Thank you. Someone with a calendar who's not calendar challenged like I am. Okay, May 8th. I'll send it out by BSpace. And where's it at, do you know? Not here. It's not here. But I'll let you guys know by BSpace and give you some warning over classes. The other thing is, for my part of the exam, obviously there are multiple choice questions, but just to let you know, under no circumstance will any of my questions have an answer like none of the above, all the above, every third Tuesday except in May or something like that. So my questions will have one unambiguous answer. Okay? And none of them will be none of the above. And at that Q&A session beforehand, I'll send out some practice questions, which are the ones I basically decided not to use for the exam, but very similar to the ones that will be in it. Okay? So I know that's coming up. I know it's on your minds. So just to let you know, it's on our minds too. Or my mind, at least, anyway. Today, we'll start kind of merging the concepts of human reproduction with development. And so we'll be discussing cleavage reactions today, which takes place right after fertilization. But the reader pages are a little scattered, 46, 58, 61, because the reader's set up for human reproduction, then development. But I've decided to talk about some of the concepts interchangeably. Then we'll talk about the blastocyst implantation, 47, twinning. We'll talk about a topic near and dear to my heart because my 3rd and 4th kids are twins. And I know what that's about from a post-birth point of view. Pregnancy and labor hormones. There's questions on page 51. Are you guys doing the questions in the reader as we go along here? You should definitely do that. And then we'll spend the last 20 minutes or so on stem cells, do some discussion. And that clearly relates to early development in humans. And we'll talk about different types of stem cells and so forth. Okay? So, brief review. For the female reproductive system, there's a bunch of cells we talked about. You know, the primordial stem cells, the Algonia, are true diploids, 46 chromosomes, 2C DNA. Females are born with about 1 million primary oocytes, which undergo the first DNA duplication. And by puberty, there's about 400,000 left. Most women in their lifetime use up maybe 800 or so of these, maybe. So you've got more than enough. At ovulation, you get the secondary oocyte plus a polar body, and then the DNA and chromosome levels are up there. And then after fertilization, you get an ovum nucleus and a polar body. Females do not actually have a separate gamete that's haploid. Females have a gamete-type nucleus. The reason being the sperm nucleus is already in there. And then you have a zygote, which is back to a true diploid. You should know the events and know the comparison to the male system. Then we talk about follicle development, which is obviously specific for females, with a little bit of overlap with the males, in that some of the hormones are similar, some of the steroids that are produced are similar, not all of them, but some of them. So you have to know what the primary oocyte does, the zona pellucida, the granulosa cells, which makes estrogen from testosterone, theca cells, that makes testosterone from cholesterol, the antrum, ovulation events, and the corpus luteum, the fact that the corpus luteum makes estrogen plus progesterone. Very important issue in understanding the menstrual cycle. Okay? So in the menstrual cycle itself, one aspect of it that I stress are the ovarian steroids. During the follicular phase, which is the first 14 days, you go from low to high estrogen. Then at ovulation, you have the corpus luteum, so you have estrogen plus progesterone. These are very different hormonal situations. Low estrogen, you have negative feedback. High estrogen, positive feedback, and an LH surge, which causes the LH surge. And after ovulation, you have inhibition. And those are different situations. So in terms of the negative feedback, hypothalamus makes DNRH, which we've talked about many times. It's the anterior pituitary. You make LH and FSH. Theca cells, granulosa cells, ending up with low levels of estrogen. During this phase, the first part of the follicular phase, you make a little bit of estrogen. The estrogen then has a negative effect on the hypothalamus and anterior pituitary. Estrogen levels start coming down. When it's too low, these negative graphs here, these negative parts of the pathway disappear. So then you start making more estrogen, it comes back up. You have too much, you now kick on the negative part of this, of the feedback, and it comes back down. So you're trying to keep estrogen within a constant level by negative feedback. However, during this time, there's proliferation of the granulosa cells, the theca cells, so there's a lot more cells. Therefore, there is a lot more estrogen. So the system is trying very hard to keep this negative feedback, keeping it constant. And you finally cross a threshold where all of a sudden you kick into positive feedback because the estrogen levels are too high. And in positive feedback, more estrogen gives you more LH, FSH, DNRH, which gives you even higher estrogen, which then eventually kicks on an enormous surge in LH. And it's that surge in LH that kicks on ovulation. There's also a smaller surge in FSH, but it's thought that the surge in LH is the more important. Okay? Any questions on that? The surge in LH is one of the triggers for ovulation. Okay? Then after ovulation, the remaining granulosa and theca cells become the corpus luteum, where estrogen plus progesterone is made, and this acts as an inhibition. Just clamps down on the anterior pituitary, clamps down on the hypothalamus. You have virtually no GnRH, no LH, or very little FSH and LH. Okay? The reason being, you maintain estrogen plus progesterone throughout the entire pregnancy period. Not by the corpus luteum necessarily, and you'll see that today, but you maintain high levels of E plus P the entire pregnancy period, which prevents follicle development. So you don't want another follicle developed during pregnancy. Yes, question? I'm not sure of your question. Yes, there's a big surge in here, and yeah, you'll make more testosterone because the testosterone gets converted into estrogen. I already have it up there. So yes, both of these take place. Because, well, you have enough. Yes, you get a little surge in FSH. But the surge in LH is not to make a ton of testosterone to a ton of estrogen. The surge in LH has a more direct effect on the ovary and triggers the ovulation. That has another role there. Very good question. But basically, view this as a unit. That both these hormones are produced during the positive feedback, make a lot more testosterone, a lot more estrogen, which then makes more of that, of the LH and FSH. That's a classic example of a positive feedback system. Okay? And as we talked about, the main components, or the main mechanistic components of the female contraceptives is estrogen plus progesterone. Okay? And as we talked about, yes, Alex, right? Is that your name? What is the function of the polar body? Nothing. The thought is that the polar body eventually gets disintegrated or degraded. What it does is you have unequal cytokinesis. So the only role is, in males, remember you get one spermatogonia, makes a primary spermatocyte, 2 secondary oocytes, 4 spermatids, right? Equal distribution of cytoplasm. The females, all the cytoplasm, or virtually all of it, goes to the oocytes. And eventually the ovum nucleus becomes the zygote. So the only role of the polar body is to supply that extra cytoplasm. So it's just really a nucleus that's left over. Okay? Okay. And so anyway, so this is, and the fertility drugs, the basis are LH and FSH, or drugs that stimulate that. Okay? So then we talked about the last bit of this, in the ovary, follicular development will continue today. It develops, the secondary oocyte comes out, forms, gets taken up by the fallopian tubes. Know the fertilization events, ion changes, cortical reaction, so forth. There's a metabolic upheaval. And this metabolic upheaval causes a last meiotic division, the second one. So then you're sitting with an ovum nucleus and a sperm nucleus. Then the nuclear envelopes degrade, the DNA recombine to form a new nucleus, and you get your zygote. That's what we ended with. Okay? Now, one thing I just wanted to start out, I usually like to do the end of the lecture, but I'll start out this time, a political issue, and that is in Arizona, some of the lawmakers there wanted to say development of the primary follicle is the start of life. What is the DNA content of the primary oocyte? What's the DNA content? 46 chromosomes and how much DNA? 4C, right. So according to Arizona, life is 46 chromosomes, 4C DNA, which we know to be virtually impossible, okay, at least the way we know it. So the point is, politics get in the middle of a lot of this, and we'll hit this with stem cells also. This is why it's important to discuss these issues in a class like this. Okay? Any questions on that where we ended on Friday? All right. So after the zygote happens, there's a series of cleavage reactions. And I'll talk about this in a little bit more detail. There's a 2-cell stage, a 4-cell, and I'm going to drop the 8 cells. You get an 8-cell morula. Morula just means ball of cells. These particular cells are all considered identical. Not considered, are identical, up to the 8-cell morula. And as we talk about stem cells a little later in the lecture, the source of totipotent stem cells are up to this 8-cell stage. Very important issue. So these cleavage reactions, so here's the zygote nucleus. You get this 2-cell state. Let me just draw this a little differently here. So here's the zygote with the zona pellucida, ZP and zona pellucida. The zona pellucida hangs around for a little bit. And what happens is, in the cleavage reactions, you have a partition in the cytoplasm. So you're getting more cytoplasm, basically you get smaller cells, but the nucleus undergoes mitosis. And so at the 2-cell stage, you have 2 cells, 2 nuclei, put the nuclei in there, 46 chromosomes, 2 cDNA, but there's half the cytoplasm. And the same thing happens at the 4-cell stage. The zona pellucida is still there. It eventually hangs around for a while. I have no idea if I do this exactly right, but the point is, again, you're partitioning of the cytoplasm. These cells up to the 8-cell morula are completely identical. And the term given to these cells are called blastomeres. So for the cleavage reactions up to the 8-cell stage, the 8 blastomeres are completely identical. And as you'll see, it's the source of identical twins. Is up to about the 8-cell stage, true identical twins. Okay? So this cleavage reaction takes place, and you get to this 8-cell morula in about 4 days after fertilization. And the next thing that takes place is a phenomenon called compaction. So you have a compacted 8-cell morula. And what's happening in compaction is you have cell-cell adhesion. And the cells just come together and are more tightly attached to each other. And remember, we talked about adhesion molecules for adhesion junctions. You can remember way back in the first lecture in the series. Can you name me an important molecule in cell adhesion? You can look at your notes if you want. Yeah, you have cadherins. And it's the cadherin molecules really play a major role in compaction. Here's an example. We can have cell adhesion unrelated to a junction, but very important. So let me just send this back down. This is the endometrial lining. Endometrial lining. And as we talked about with the menstrual cycle, the optimal implantation of the endometrial lining is when? It's about 7 days after ovulation, right? Remember that? We had that on there? And it takes about 7 days to eventually implant something. And what you implant is something called a blastocyst. And a blastocyst has a part of it with cells in it called the inner cell mass. And inside, as part of this process, you build up fluid. So inside is something called a blastocele, which has fluid in it. And on the outside, you have something called a trophoblast, which is part of what implants, important part of implants. And this inner cell, this blastocyst, occurs at about day 7. So from day 4, you go from a compacted morula of 8 cells to a blastocyst of about 100 cells. Okay? So this is the structure that's getting ready to implant. Yes? What's the difference between compaction and implantation? Totally different. Compaction happens up here. They're just the cells getting tighter with each other. Implantation is you're implanting in the endometrium lining at about day 8 or 9. So what happens is this structure implants. And it's the trophoblast interacting with the lining of the endometrium. But now this inner cell mass is now, let me put this over here. What implants is the inner cell mass actually differentiates a little bit. So here's what implants. So what this is right here is now the epiblast, which becomes the embryo. And we'll see that a little bit later on, probably Wednesday. And there's also inside here something called a hypoblast. A hypoblast is an evolutionary artifact in that it's actually the equivalent of the yolk sac. Humans, mammals don't have yolk. But it is an evolutionary artifact. But what it makes is the initial red blood cells to kind of get the circulation system going. And this is what implants here at about day, so epiblast plus hypoblast. This is at about day 8 to 9 when we start getting this implantation. So let's look at the sequence of events. Zygote to 8-cell morula takes about 4 days. All 8 cells are indistinguishable. Very important to remember. There's a compaction due to adhesion molecules. The molecules that come together, the cells come together, tighter interactions. And then from day 4 to about day 7, you form a blastocyst, which is comprised of the inner cell mass, which will be important in our stem cell discussion. There's fluid in the middle called a blastocele and a trophoblast around the other outside. The trophoblast is considered an extra embryonic tissue. So the embryo, the trophoblast does not become part of the embryo. Upon implantation, the inner cell mass becomes the epiblast and the hypoblast. And it's the epiblast that becomes the embryo. Again, we'll talk a little bit more about that Wednesday. Okay? Any questions at that point? Yes? I'm not sure of your question, but this is the endometrial lining of the uterus. This is where it has to implant. So the uterus. Okay? There's another question somewhere I saw. Okay? All right. Shout it out. Where are you? Oh, right there. We'll get to twinning. But identical twins come up to the 8-cell Marula stage. Yeah, we'll talk about that in a matter of maybe 5 minutes, maybe 10. Yes. Yes, the trophoblast has a function. It eventually becomes the chorion, but we'll talk about that. It also gets involved in implantation. Now, when it implants, you start making the placenta. The placenta, which is composed of maternal plus fetal tissue. Secondly, we'll continue over here. The placenta does another very important thing. The placenta makes an important hormone called HCG. Here's the beginning of it. It's one of the first events that takes place. And this is human chorionic gonadotropin. Okay? What this does is this maintains the corpus luteum. What does the corpus luteum make? Estrogen plus progesterone. You need, as you'll see in a little bit, estrogen plus progesterone during pregnancy. So for the first part of the pregnancy, the placenta makes human chorionic gonadotropin to maintain the corpus luteum. And this occurs soon after the formation of the placenta. And, therefore not surprisingly, HCG, I'll put this board down in a second, is the basis of the pregnancy tests. So right after implantation, which is, think about it, day 8 or 9 after ovulation, within a day or two, placenta starts making HCG. And then this is the antibody detection of HCG is the basis of the pregnancy test. It is also, this is important, you can discuss with your moms, this is also the basis of morning sickness when a woman is pregnant. Okay? And if you have twins, or if you're a member of a twin, there's extra HCG around, and there's extra morning sickness many times. Okay? If there's a level of it, you can ask your mom, I know you're part of a twin. You can ask your moms about that one. HCG only hangs around during the first trimester, and you'll see that. That is why the morning sickness goes away at the end of the first trimester. We'll talk a little about that more specifically. But the point is, HCG does a bunch of stuff. But importantly, it maintains the corpus luteum. Okay? And as we talked about, keeping this implanted is a progesterone receptor-dependent process. And therefore, the abortion pill is a progesterone receptor antagonist. And we talked about that when we talked about hormones. This is where it fits in right here. Okay? Questions on this? As we start talking about twinning. Okay, so I'll keep this part of it up. Just so you should know the process, the order of events that takes place. And for twinning, I think we'll go up here. Now, there are different kinds of twins. One kind of twin is called fraternal. Fraternal twins have 2 distinct fertilizations. And therefore, 2 zygotes, 2 distinct zygotes. So in the case of fraternal twins, as you can imagine, there is more than one secondary oocyte that becomes available. And it gets produced, and both of them get fertilized. Okay? A lot of times women on fertility drugs will kick out extra eggs, or extra secondary oocytes, and therefore they're susceptible to not only twinning, but having triplets, quadruplets, quintuplets, etc. Okay? In fraternal twins, they're exactly like any siblings. So my set of twins are a boy and a girl. And so they're obviously siblings. Okay? They're not identical. They're genetically like brothers and sisters, as opposed to anything else. Okay? Secondly, it is possible. There are examples where somebody gets pregnant, goes on to keeping this blastocyst going, and for whatever reason, a month or 2 later, another secondary oocyte can come out. And they can have twins that are actually born several months apart, or several months in difference in age. It's rare, but it does happen. And those would also be probably considered fraternal twins. Maybe, maybe not, depending on one's point of view. In this case, we'll just only consider, I will not ask you about that in any exam. But just fraternal twins are 2 distinct fertilizations. Okay? This is to be distinguished from identical twins, which is 1 fertilization, 1 zygote, 1 fertilization and 1 zygote. And then somewhere up to the 8-cell Marula, the cells during cleavage come apart, separate. So as I said, all the cells up to the 8-cell Marula are completely identical. So if one of these comes off, if one of these cells comes off, you can then continue the developmental process. Okay? Most identical twins, the majority of them, split off at the 2-cell stage. So you have a zygote, you have a 2-cell stage, then these split. And the zygote and the 2-cell stage, the 4-cell stage, 8-cell stage are all identical. So then these guys then develop from 2-cell stage, 4-cell, and the 8-cell Marula. But these are absolutely identical at this stage. And they are born absolutely identical. Okay? Note the distinction between an identical twin and a fraternal twin. Now, there are variations of this theme. There's another phenomenon called mirror image twins. Mirror image twins are genetically identical, but they split later on.